Of course, if we only had short-term plasticity mechanisms, we wouldn't be able to learn or adapt on longer timescales. You wouldn't, for instance, be able to learn to ride a bike, or remember your own name, or learn anything from watching this video. In fact, even simple organisms like flies, which exhibit a relatively limited repertoire of relatively hardwired behaviors, exhibit significant amounts of learning in response to a changing external world. So how does this work? In order to be able to store information in the nervous system long term, we need to have mechanisms that can alter synapses in a stable way that lasts for a long time, possibly even over the timescale of the entire lifetime of the organism. Two well-described phenomena for achieving such plasticity are so-called long-term potentiation, or LTP, and long-term depression, or LTD. As the names might suggest, long-term potentiation makes synapses stronger, and long-term depression is a process that makes synapses weaker. The phenomenon of long-term potentiation was first described in 1966, when researchers in Norway conducted experiments at specific synapses in the hippocampus of anesthetized rabbits. In particular, they found that when they strongly stimulated a presynaptic fiber, resulting in a strong burst of action potentials, the effect of subsequent stimulation of those same fibers on the postsynaptic neuron was potentiated, or strengthened. Even more surprisingly, the potentiation lasted for a long time, up to hours. Stimulating those same fibers at moderate levels didn't have this effect, and in fact, if the fibers were stimulated at a low, infrequent level, the synaptic strength would gradually decrease. Not surprisingly, the phenomenon of LTP and LTD instantly became a subject of a great deal of scientific interest, since they were for the first time suggested a mechanism by which memories might be stored. Since the original discovery of LTP in the hippocampus, LTP and LTD have been observed in many other neuronal structures, including the cerebral cortex, cerebellum, amygdala, and many others. Some scientists have suggested that LTP may indeed occur at every excitatory synapse, everywhere in the mammalian brain. Over the next few segments, we'll explore what is known about the mechanisms of LTP and LTD in depth.